Okay, thank you very much for the kind introduction, and thank you for inviting me to give you this presentation. So, I know that we have, it's an early start after last night's fantastic dinner, and I know there was no coffee at breakfast, so I understand why some people may be feeling a little bit, soft around the edges. But without further ado, what I'm going to try and do is not go into too much detail, but to give you an overview of how we are now trying to really drive everything down to a genome-resolved level of interpretation of metagenomics. So I am agnostic in terms of microbiome that I'll study, so I'm jack of all trades, master of none. So we look at all sorts of different environments, but I'm going to start off with the human microbiome, and specifically the human gut. And it's quite clear that the human gut has been highly implicated in both health and disease. And when we take health, it's all to do with dealing with food and nutrition. And disease, we're thinking about things like inflammatory bowel disease and so forth. Within our human gut, we know that there are thousands of different bacterial species and strains. And we know that they are very different as we move from person to person. So where you and I are probably 99.9% genetically identical, our gut microbiomes can differ as much as 80 or 90%. The difference comes from all sorts of things, such as if you've taken a course of antibiotics, food you've had or not had in the case of coffee, if you're taking other medicines. And those medicines can also be metabolized by the gut microbiome, and it has another effect. And all of these factors, we're only just beginning to understand. And so we start off in the gut microbiome pretty much because it's easy. It's a high biomass environment. It's very easy to get access to the stool sample, which we use as a proxy to the gut, and it's not too complex. So the real key grand challenges for human microbiome research I put them as these 4 levels. The first is understanding the diversity of that microbial composition. And we are going to talk a lot today about species level, but strains is becoming more and more important. Then we want to start understanding the interactions between those players in the microbiota. But we need to know who's there and when we need to get to them. And only then can we start actually having, and this is the promise of the microbiome, of actually investigating how we can start manipulating a microbiome and modulating the interactions. So my work started in terms of the human microbiome and gut about 5 years ago, when there were a number of papers dealing with culturomics. And then every single paper would come out and say, oh, we've cultured 800 new bacteria from the gut microbiome. And so there are a whole range of these, and one of these papers came from Trevor Lawley, who's based in the institute next to the European Bioinformatics Institute. And he was like, yeah, we're getting there. I think we'll have this nailed really soon. And I was saying to him, but when we look at metagenomics data, those isolate genomes can only really account for a small fraction of the total data set. And he was like, yeah. You know the sort of typical coffee. So he said, well, why do you, we knew that assembly was getting better and the techniques were getting better to resolve. So he said, okay, let's go and see what we can do at scale. So here is my sort of workflow diagram, and I just really wanted to emphasize these are the things that we do. And we very much are about making sure this data is archived appropriately and is available in the resource called Magnify. So on the far left-hand side, you start off with your raw shotgun metagenomics read. We can just analyze those reads, but they're all fragmented, and it doesn't really give you so much information. What we've really started doing is making sure we do assembly, so we assemble those reads. We then push those back into the European Nucleotide Archive. We can then annotate those, and we get access to lots and lots of proteins. For example, Magnify now sits on a 2.8 billion non-redundant proteins. That's quite big. But what we're doing more and more in this terms of trying to get to the genome is doing this process of grouping contexts together we think have come from a single genome in a process called binning. We'll then do quality estimation, and we do this using a number of different approaches. One is to use CheckM, which uses single copy marker genes. The other's another tool called GUNC, uses compositional analysis, and we're trying to work out whether that grouping really reflects a genome and how well. We may do some refinement process. And then we have a set of genomes, which we refer to as MAGs, metagenomes assembled genomes. We'll do taxonomy. We push those also into ENA. One of my proudest achievements is I've broken INSDC by the amount of data I've submitted in, and so we had to back that out. There is now a special layer within the European Nucleotide Archive for collecting all of this data. And now what we're doing is trying to remove redundancy because we see the same genome over and over again and have biome-specific catalogs. And we've done that for the human, and we started off with the human gut. So when we were doing this, one of our approaches of verifying the quality of these MAGs, because this is one of the key questions, is am I generating an arbitrary genome of all chimeric sequences, or am I actually getting something correct? So this large collection of isolate genomes has really helped us. So what we could do is take these de novo MAGs, and so they've not seen the isolate genomes at all, and then we can compare those. So on the far left-hand side, what we have is the fraction of the MAG aligned to an isolate genome, or the reference. And anything that's more than 60% aligned is a good overall alignment. And that's illustrated with the green dots. So we have on the top side here, this is all the things that match an isolate. The grey dots are things that don't match. If we then zoom in and look at the average nucleotide identity in that top range of 60% or more, the dark blue dots are those that have greater than 99% ANI. So this is where we're actually reflecting probably strain level. We must remember a MAG is always a population genome, but this is very close to what a strain looks like. And so what we have with these dark blue dots is they're all pushing to that top right-hand corner where you're getting greater fraction of alignment. At a lower level, at 95, the lighter pale blue, that represents species matches, and again, they're all in that range where you would expect them. We're not the only people to have generated these MAGs for the human gut microbiome. So at the end of 2019, there were two other groups, so Nikos Kyprides at JGI, and also Nicolas Agata in Italy. And that actually gave us a way of actually independently comparing these genomes. So in this upset plot, we have the three different groups. The one where the largest bar is actually where all three agree on the genomes. The next two smaller, darker bars, skipping one over, also where two of the groups agree. This one large set from the Sagata group, actually, these ones where they were novel. Upon further investigation, this is because he had some novel data sets of hunter-gatherer populations that just were quite unique and we hadn't seen, and that's where that extra set comes from. So what we see is this is quite reproducible. So overall, we now have a unified human gut catalog. We started off with our 12,000 shotgun metagenomics data sets. We went through the assembly process that I've already outlined and identified nearly 2,000 species. Merging all of those catalogs together has given us nearly 5,000 species. On the right-hand side is a taxonomic tree of those. And what you might not see the finer details on this, but if I can just draw your attention to what the colors coming off the tips of the branches, the green are those where they're uncultured. These are MAGs. If we find an isolate there, it will be colored either red or blue. And so what you can see here is that there's a vast collection of genomes that are yet to be isolated. And those that have been isolated from the human gut are really unevenly distributed around this taxonomic tree. So this gives us a really good overview. I would illustrate, although we're talking about 5,000 species representatives, that actually correlates to 210,000 non-redundant genomes in that catalog and 170 million proteins. Very briefly, why do we have so many of these uncultured? We started looking at the metabolic potential of these and what they can metabolize. And we think that actually one reason is if we take different phylogenetic groups and just look at their functions and look at the effect plot, there are a whole bunch of processes and metabolic functions that are involved with dealing with oxygen tolerance that are just absent from those MAGs. So we actually think that a lot of the reason for these uncultured species is that they're really sensitive to oxygen, and when you take a stool sample, it gets exposed to air, and this actually kills them, and then actually this becomes very hard for you to actually then recover those in the lab. So we've also looked what else is in these metagenomic data sets. So we started off with the bacteria. So the next thing is we started looking at the viral composition. Now, this viral composition is purely skewed to DNA viruses. So we took the isolate genomes from the Lawley lab and our set of metagenomes that had been expanded. And then we used a number of tools, VirFinder and VirSorter, and actually we came up with a new pipeline called Virify, which uses VirFinder, VirSorter, and PPR Meta now to try and sift through all the contigs to work out which contigs look like they're viral or which contigs have a viral sequence embedded in them. What's unique about Virify is we have a set of diagnostic profile HMMs to try and assign taxonomy. The reason why that's unique and important is there are just so many viral sequences out there. So I'll come on to that in a second, but we used this initial process of trying to sort through. We used very strict length filtering and developed a neural network to try and work out when things are actually viral versus not, or because a virus quite often looks like a plasmid, and a plasmid, etc., is confusing with other parts of the microbiome. But we came up with the gut-phage database that has 142,000 quasi-species in it, of which a large fraction of those just have no taxonomic annotation. However, what we can do is look at the distribution of those phages. So a well-known crass phage was the most abundant, but there was another phage called the goobophage, which was also highly abundant. So we identified this goobophage in over 650 different genomes, but yet no one had identified this before. If we look at where those genomes have come from, they have a broad geographical distribution. However, there is a bias for those to be in Europe. So when we actually normalize for actually the numbers of samples, that actually the European population has more, and actually the US population, or North America, actually has less. The rest of the countries, it's about what you would expect proportionally. The goobophage is related to the crass phage. It's phylogenetically distinct. And so the key thing here is that until we applied this metagenomics approach, this widespread goobophage has been sitting in our microbiomes, yet nobody has realized it's there. Okay, so we've done viruses, very, very quickly admit. What about eukaryotic genomes? So I am very much of an approach of I don't like anything complicated. So we wanted to start understanding, can we recover eukaryotic genomes? And actually, so we started off with a very simple example. Something that's quite well understood is kombucha. It's one of these fermented teas. And it has one dominant eukaryotic genome and a range of bacterial genomes. So in this particular example is actually a GC content of that kombucha sample with the reads that align to the well-known eukaryotic gene, this penicillium, actually colored. And there's two colors here, the red and the blue. That is because when we go through the binning of this particular genome separates out into two distinct groupings. And if we look at that in a t-SNE plot, what we can see is actually, yes, they've got similar GC content and they are connected, although it's a long part. If we look at those two different bins, we see that they're evenly distributed across the whole genome. So we've seen this over and over again, and we have a tool called eucc. Now, eucc was initially designed for estimating completeness of a eukaryotic genome. It works in a similar way of using single copy marker genes, but for eukaryotes it's much harder because of the fact that there isn't a set of universal single copy marker genes that can cover the genome. So eucc does a double pass. So it initially works out what it thinks a taxonomy is, and then picks out a clade-specific set of marker genes. When we were running this, we saw that Kombucha example over and over again across different data sets. So we thought, actually, we're quite often seeing the same genome come up. One bin looks fairly complete, and then another bin is quite incomplete. And so we wanted to start coming up with a method for actually trying to work out when a genome is being split across two bins. And so we've extended eucc to actually detect this and come up with this notion of merged mags, or M-mags. And we'll only do that in a very conservative way, so it has to increase the completeness by more than 10% without having any impact on contamination over that, on that QS50 score. So what we do is we take a primary bin, we do this initial pass of trying to estimate that completeness and contamination, and then we look through all the remaining bins that are below that 50% complete and look for matching paired end reads to then give us evidence that these things should be joined. And we look for more than 100 paired end reads. It's a bit of an arbitrary figure, but they have to be at the ends of the contigs between the two bins, and then we'll bring a merge bin, and we've done that. We can then use that and align those against reference genomes or the best quality and produce these alignment fraction plots and coverage and ANI. And what we find is that when we compare the primary bin to the secondary bin, the ANI against the reference and the ANI of the secondary bin compared to the reference of the ANI of the primary bin compared to the reference and the ANI of the secondary perfectly correlates. So it really gives that strong suggestion that they should be related. Another example, and this is really interesting, is so we applied this to skin at scale. We have 13 merged mags, all belonging to Malassezia globosa. And what we can see, these are all aligned to that reference. And actually, the blue is the primary, yellow is the secondary. And actually, you see, although these are completely independent sets, you see the pattern of this primary and secondary binning is very, very similar across those different genomes. What we did also see is that they both had two regions where there were gaps compared to that reference. And we were really interested to know what they were in a little bit of detail. One of these, consistently, there were paired ends that just bridge across here, and all of those 13 merged mags compared to the isolate. So this looks like an insert in the isolate compared to those merged mags, because we've got paired end information suggesting these should be really close. The other gap corresponds to the ribosomal region, and it's a well-known region that is just really hard to assemble out using short reads. So actually, we were really happy with this. So it really suggests that there is compositional bias that causes this split binning, but we can recover these to get eukaryotic genomes. In the human skin microbiome, there are far more eukaryotes that are far more accessible and have got lots of cultured isolates. On the left-hand side, red are the cultured isolates, blue are the mags that we've recovered from the skin de novo. Some match perfectly, so where you see a straight line in the tree, or vertical line, that represents a perfect match. We found 3 novel mags sitting on the skin. And if we look at those on this second graph, where we see we've got body site and which healthy volunteer they've come from, and then the genomes along the x-axis, what we see is that one is really, really common, and that's been called Malassezia oris because it's normally found near the ear. And then there's Malassezia palma, found on the hands and feet, and then Malassezia rara is actually quite restricted and is only found on a few body sites on one individual. We've also done this across a whole number of assemblies in Magnify. So we scanned nearly 20,000 assemblies for potential eukaryotic composition. That's a lot of work to get. So we came up with 751 eukaryotic genomes corresponding to 124 species, of which 58 are novel, across a range of different environments spanning aquatic, engineered, human gut, skin, lichens, etc. And they have a broad taxonomic distribution. So we are now able to recover eukaryotes. These have a distinct metabolic function. So we shouldn't, although they're low in numbers, they have a distinct metabolic function. So in here we've got both KEGG and KZ pathways. Anything that's in the third column is unique. And if we look at, for example, in the lichens, they provide a huge amount of metabolic function to that lichen that is absent from the prokaryotic fraction. We've applied these approaches to other environments beyond human. We've got a chicken secum catalog. Carsten is also on that project. There we have 825 species. We do pick up archaea. They're not shown in this tree. 570 of those are novel and don't match anything. We get one blastocyst, so a eukaryotic, and thousands of phages. And we're now doing this routinely. So finally, this is really just a quick advert that Magnify, the database I run at EBI, is collecting these catalogs. We have 4 at the moment, and they're growing in terms of numbers. We've updated the human gut catalog such that that now has over 5,000 species in there. So the ones that we pull out is really based on fundings and biomes of interest. This data really is drawn from this special layer in the European Nucleotide Archive. And I would emphasize, if you're generating mags, please put them in there, because we can then capture them, annotate them, and then put them out there. Really quick take-home points is, metagenomics is just revolutionizing what we understand of the whole microbiome. And it is possible now to get complete genomes. We can get very close representatives of isolate genomes, really confirming the approach, and we find those consistently. Given enough data, and the sample extraction hasn't excluded it, we can get eukaryotic genomes, and the viral diversity is just crazy. And now the next step is, now we've got these catalogs, is to really start understanding those functions they encode and how they actually interact with each other and the host. And thank you for listening. And I'll thank all of these people, and if there is time, I'll answer any questions. Thank you.
Thank you, Rob. So I think it's excellently you put points on these different aspects which all should care using the metagenomic approaches. Please, yeah. No, the first one. Yeah.
How is 80 to 90% difference defined? Because if it sort of incorporates frequencies or presence or absence, would give you completely different results.
Yeah, so that 80 to 90% is at the strain level. If you take an individual, me compared to you, at a presence or absence point.
If you define as a number of reads belonging to a specific species as a multidimensional vector, you get a different result than if you say presence absence, because you can have 90% of the reads being common, and it's just rare things which are different.
Even things like uniformis and vulgaris, which are in pretty much everyone's microbiome, if you look at that composition from individual to individual, the number of reads that map varies massively to sort of 10 to 15% of all reads can map to those 2 genomes in some individuals, and that can then really tail down to looking like something really sparse. So I think the take-home message is that it's both really variable, both on composition and then abundance. is also highly variable, and I don't think we know exactly that pattern and what that means.
Okay. Just very quickly, for how many could you make metabolic models analogous to the E. coli? For how
Okay,
many of your species?
so you're fraught with danger here, because all of the metabolic modeling is based on E. coli. These things look really, really different to E. coli. So even E. coli, we only know about 70% of the functions of E. coli at best, if you're being nice. These genomes, less than, well, 50% have no known function at all. Of 50% of the coding regions have no function assigned to them whatsoever. So making a metabolic model is only going to be using a fraction of the data, and then you're using a proxy to E. coli. Now, one of the things we have going on at EMBL, and this is not in my group, but across EMBL, is actually turning some of the, so Vulgatus and Uniformis is actually developing those into model organisms and actually understanding them completely like we do E. coli.
Okay, and then one more question from Duskaya. Maybe you can take that.
Well, thank you very much, Robert. It's great. We all love and magnify. We use them. I have a comment or two.
Okay,
nevertheless.
thank you.
One is, you know, when we think about genomes in terms of species, then we think about the genes that are common to species, a core genome. We think about accessory genes, and we think about pangenomes.
Yep.
And this is kind of lost when you guys, you know, suggest a collection, non-redundant collection of genomes. So my comment is, you know, maybe we should try to get together and think about how to capture all that. And before I end, just, you know, for historical accuracy reasons, the first metagenomically assembled genomes were published in a paper of 2014 by Mathieu Almeida, who is sitting in the audience. And, you know, it's a shame that people don't acknowledge that precursor work, you know, and start with what happened in 2019.
So on that point, I didn't say we were the first. I think and in a 20-minute talk, I already ran out of time. So apologies. So with respect to your question about core, accessory, and pangenome, so every single one of those species representatives in Magnify, we have all that data, and that is available in terms of what we think is core and that pangenome. We are thinking very much in that, and actually going back to the question about numbers of species, there is a question of actually taking a vector of function. And I think that absolutely, the vector of function is the way that we should be considering rather than trying to pigeonhole everything into a species.